We are going to be discussing in genetics. So I hope you bring your A game this entire semester, because probably 60% of your test is going to be from genetics, ecology, and evolution, which are our 3 units this semester. And that also means since there's only 3 units, there's only 3 big tests. So yeah, we're going to be working on genetics for about a month and I've got your test down for February the 10th. So we're going to be here for a while. There are 2 distinct parts to the genetics unit. Molecular genetics, which is kind of the DNA and the basics, the small scale stuff. And then Mendelian genetics is more the Punnett squares and how stuff's inherited and pedigrees and stuff like that. So that's kind of how we'll split this up. And we'll probably have a quiz about halfway through once we get through with the molecular genetics, just to see where you are. I think that's probably the more difficult part, just because there's so many details that you guys probably are not familiar with. But I am sure you are all competent enough and diligent enough to do well in this unit if you will pay attention and do what you're supposed to. Walker asked a very important question. Are you doing the same thing on Thursday and Friday as you are on Tuesday and Wednesday? And the answer is no. Thursday and Friday, we will be working on protein synthesis, parts 1 and 2. And if you are at home, it's important that you are working through the first 2 sections on history, structure, and replication. And then, because otherwise, when y'all get in here on Thursday and Friday, the 3 of you who are listening to this recording are not going to have any idea what to do when it comes to protein synthesis. And the same way for you guys here, if you don't pay attention today and tomorrow and understand what's going on, protein synthesis on Thursday and Friday is not going to make a lot of sense. Y'all are going to be working on notes, reading through and looking over those, and I've included a couple of videos on protein synthesis for you guys so that you don't get behind. And I think Mr. Bozeman and Crash Course is probably going to do a good job of explaining all of that. And then Tuesday of next week, hopefully we're all back together. We'll be working on a protein synthesis lab, which will take most of the day. And hopefully it'll kind of tie everything together. And if you're out, those of you guys that are out on Thursday and Friday, if you're having trouble, then we'll sort everything out on Monday and Tuesday, hopefully. I'm hopeful we don't go A and B next week. Yes, ma'am. I have prepared, and I think we're going to do a mock exam on roughly April the 12th. So we're running a little bit behind, but we'll see somewhere in there. The mock exam is going to be your final exam. I'm not going to do it on a Saturday. My plan is we're going to have a multiple choice section, which will just be an entire class period, probably about 50, 60 questions. And then the 2 following days, we'll split up and do FRQs. And then, hold on a second. So, we'll split up and do a couple FRQs the 2 following days, and that will be our mock exam. We're not going to do a traditional one where y'all come in and take the whole test at one time. I would have to have too many people in the library, and we don't have a day off like we traditionally do, and I'm not coming up here on Saturday to do that. So we're going to use our time in class, and we're just going to kind of break it up, and then we'll work through and grade it. And go over it, and then we'll spend 2 or 3 weeks reviewing. Hopefully, you guys will be well prepared by that point and ready for the test, which is on, for those of you who have calendars, your AP exam is on May the 14th at 8 a.m. So I think you guys will have plenty of time to be well prepared by that point. And I know that as significantly difficult as this class is, that if you're doing what you're supposed to in here, then you should have a good opportunity to do well in the AP exam. I feel confident. Hey, come here. No running. Leave that door cracked. Okay. Any other questions for this semester? We're really going to get in the meat and potatoes of probably what you're going to see on the exam. I would say 40-50% of your FRQs on the exams usually wind up being genetic stuff. So we're going to do a lot of FRQs as we can fit them in, and then hopefully, you guys will tie, all this will tie together and fit together with things we've been talking about, and then genetics will come. All of this will tie back into evolution in the last unit, because evolution is all about changes in genetics is really what it is. And there's a lot of math that goes along with it too. So anyway, that's where we are. That's what you need to kind of prepare yourself for mentally. On page 313 at the top of the page, let's go through and look at some of these experiments that you need to be familiar with and how we got to where we are in terms of what we know about genetics. We'll talk about COVID quite a bit more. We'll talk about the vaccine. And hopefully we'll kind of alleviate some of your fears if you have crazy people in your family like I do that think the vaccine contains nanobots that are going to upload data to your phone, and in order for you to buy and sell and exchange goods. So I think the vaccine is actually pretty simple and straightforward compared to a lot of vaccines, especially with your knowledge of immunology. It shouldn't be that hard to understand how it works. So anyway, everybody gets to make their own choices though. What do we look like? We're in communist Russia. All right. Important concepts from previous units. What are the monomers of nucleic acids? Here, better question. What are the two types of nucleic acids? RNA and DNA. So those are the two types of nucleic acid. Remember, you got your 4 macromolecules: proteins, lipids, nucleic acids, and carbohydrates. So the two types of nucleic acids are DNA and RNA, and the monomers of a nucleic acid, or those nucleic acids, are Nathan? What is the monomer of DNA and RNA? Ashlyn? Nucleotides. What are the 3 components of a nucleotide? So remember, if DNA is a train, the cars that are making it up are nucleotides. And the nucleotides, what are they made of, Tay? What? Nitrogen what? Nitrogen base. You're close. Are y'all got the notes here? Number 1, the basic unit of DNA or RNA is a nucleotide composed of a nitrogen base. That's your A, T, C, and G, adenine, guanine, cytosine, thymine. Your 5-carbon sugar, in the case of DNA, it would be... No. 5-carbon sugar in DNA is... What does the D stand for? Oh my gosh, y'all are all going to fail. Deoxyribose. So the sugar is deoxyribose. Remember, sugar's in an OSC. And then you have a phosphate group. So what of those 3 parts makes up the backbone of DNA? So if DNA is a ladder, what's the outside of the part of the ladder? Phosphate? And the sugar. Good. And what makes up the steps in the ladder? The nitrogen bases. Good. So I'm assuming that y'all either learned nothing in 9th grade or you've taken mind-altering drugs since then and forgotten everything. All right, number 2. DNA is the $1 million blueprint. It is what is used throughout our lifetime. You have cells like in your neurons and muscles that you have the duration of your life, and that DNA is continually, sections of it are copied in order to make stuff, and they're used. RNA is the very cheap disposable copy, kind of like we see in the COVID vaccine, where it's used to make a protein or maybe an enzyme from that, and then it breaks down and it's recycled. So I'll give you a quick overview of the vaccines that are out there. The ones I've seen are mRNA vaccines, which basically means so DNA, what do we call a segment of DNA that codes for a protein? Segment of DNA that codes for a specific protein. Sarah? You're wearing them, Sarah. Genes, good. So genes are segments of DNA that code for a specific protein. Man, y'all must have really checked out over the break. So a segment of DNA that codes for a specific protein is called a gene. So when you copy a gene into RNA form, we call that's just an mRNA strand, and it's going to code for a protein, okay? So basically what's happening with these COVID vaccines is they've made the mRNA code for the protein that's on the outside of these COVID viruses. So remember from immunology, the outside has to trick a cell. The outside protein structure of a virus has to trick a cell in order to get inside the cell and to infect it. So basically, you get this vaccine, sorry, don't confuse that. You get this vaccine, which has this mRNA code. Your body makes the protein that would be on the outside of the virus. And then your body's like, hey, that's a foreign protein. What's that? And then it starts the immune response where, it makes immune cells and the memory cells. So it recognizes the outside of what this virus would look like. And then if you're exposed to the virus, then it can fight it off quickly because it recognizes it very quickly. Remember we talked about primary and secondary response? And then since it's an mRNA copy, that mRNA is going to break down very quickly, and it's pretty much going to be gone after that. So it's a little bit different than a lot of flu vaccines where you have a deadened or weakened form of the flu that you're injected with and your body makes a response to it. This is giving you part of that mRNA, and then you're using your cells to make part of that outer protein structure, and then your body is recognizing it as foreign and mounts an immune response. And so that's how you get your developed specific immunity to the virus, okay? So that's kind of what we're working with here. All right, Frederick Griffith, at the top of 313, he did this experiment, and I'm going to go through and kind of give you the simple version of this. Y'all can read through it on your notes there. This happened in 1928 by a British Army doctor who was studying pneumonia. And pneumonia is a bacterial infection that kills many people every year. And transformation is a term for when a bacterium is transformed or changed, usually by a change in DNA. We're going to do a transformation experiment in class in a couple weeks where you guys take some bacteria and you're going to take some plasmids that have DNA from a jellyfish that cause them to glow in the dark. And we are going to try to get those bacteria to take that DNA, those plasmids up. And if they do that and they start expressing those genes, then they will then glow in, I think you have to have the presence of UV light, but in the dark, okay? So we're going to try to do some transformation, and usually we're pretty successful, so we'll see how competent you guys are. But anyway, on 313 at the top, here's basically what happens. Griffith, he injected some mice with a harmful pathogenic bacteria. What happened to them? They died. He injected some other mice with a harmless bacterium. What happened to them? They lived. So the harmful ones killed them. The harmless bacteria he injected didn't hurt him. Next thing he did was he took those harmful bacteria, the pathogenic bacteria, he heated them up so that it killed them, and he injected them into the mice. And what happened?
The mouse lived.
The mice lived. Okay, so he killed the bacteria. So before he injected into them, and then when he did, it didn't affect him. Now, here's the important part of the experiment. He took the heat-killed harmful bacteria. So he had these harmful bacteria. He killed them with heat. Okay, he mixed them in with harmless bacteria that were still alive, and then he injected them into the mice. And what happened to the mice? They died. So why did the mice die when they only had harmless bacteria injected into them?
I guess the harmless bacteria took in the DNA of the harmful bacteria and transformed them into harmful bacteria.
Good. So that DNA from those harmful bacteria, some of that had to be taken in by some of the harmless bacteria, and their DNA was changed, and that process is called transformation. Okay? So what we're going through with these different experiments we're looking at here at the beginning is, how did we get to this point where we know that DNA is the molecule of inheritance, okay, that helps determine who we are and what we are, okay? So, Oswald Avery and Associates. So, in 1944, he was trying to figure out the instructional agent that was transforming the bacteria. It sparks a lot of controversy, this experiment. Really, the only thing you need to know is that part C. Controversy, as DNA is too simple a molecule that most scientists believe it must be proteins. So basically at this point, scientists have kind of narrowed it down to proteins and DNA, as one of those is the instructional agent for life, okay? They don't know which one it is, but if you look at DNA and then you look at proteins, proteins are usually much bigger and more complex molecules. And so the idea was, in order to create a big complex organism like us, then you need a bigger complex molecule. God bless America! Wish they'd quit doing that. I forgot I'm on the internet. Hold on a second. My composure. People outside are working on stuff.
Why'd the power go out?
I don't know. Okay. Where was I? So they're trying to figure out what the instructional agent for life is. What contains our genetic code. At the bottom of 313, so Hershey and Chase in 1952 conducted an experiment to find out. They know that bacteria are infected by these type of viruses called bacteriophages, and they look like little alien spaceships landing on the bacteria. I've told you guys in the past, bacteria and viruses have been fighting in wars with each other through the history of time. And so these viruses land on bacteria, they inject their genetic material, and then they use the bacteria to make more viruses, and they split open and spread their viruses to other bacteria, okay? So the scientists used this knowledge on page 314. Now, I read through the book this morning, and it was confusing, so we're going to read through the notes today. Number one, Miranda?
On the book?
No, in the notes. Hershey Chase experiment.
They used radioactive sulfur 35 to label the virus's protein outer capsid in one container. Remember, the amino acid cytosine contains sulfur. The radioactivity allows them to follow where the proteins go by using a
Geiger counter, yeah.
A Geiger counter is used to measure radioactivity.
Okay, so yeah, if you've ever seen one of those on TV, it's like you hold it up to something, it's like beep, 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 beep. That's a Geiger counter. It's used to measure radioactivity. Okay, so they've narrowed it down to two things, proteins or DNA. So they've used this radioactive sulfur, which they know is in proteins, on this outer coat of the virus. And so they basically made it radioactive. And then they used a radioactive phosphorus in number 2 to identify the DNA, because what does DNA have as its backbone? Phosphate. So we've got 2 radioactive markers here. One you've got with proteins, and on the outside this radioactive sulfur, and then you got radioactive phosphorus that's used to mark the DNA. Okay? And then depending on what's injected into the bacterium, then you can determine, well, did it inject the radioactive sulfur or did it inject the radioactive phosphorus? Okay? So in one of them, they've got the radioactive sulfur you see here on 314, and then in one, they've got the radioactive phosphorus that they exposed it to. Number 3, Matthew.
The radioactive viruses were then exposed to the bacteria. The viruses infected the bacteria. In the radioactive sulfur container, the radioactive sulfur did not enter the bacteria. It remained outside the bacteria. When the viruses reproduced inside the bacteria, the reproduced viruses that came out of the dead bacteria were not radioactive.
So that means if the sulfur did not enter the bacteria and the viruses that came out were not radioactive, that means that what didn't enter the bacteria? The radioactive sulfur, which was found in the... Why did they use sulfur? They used it to mark the protein. So the protein didn't enter it. In the radioactive phosphorus container, the radioactive phosphorus did enter the bacteria. Remember, the radioactive phosphorus was a part of the what? Part of the nucleotide sequence, yeah, part of the DNA. And so because that entered the bacteria, they were able to deduce that that was part of the DNA. The DNA was what was injected. It was what was used to help make more viruses. Therefore, the DNA is the instructional unit for life. Okay? This proved with 100% accuracy DNA was a transformational agent. It carries the blueprint of information from one life to the next. Now, we have another experiment here that helps us understand DNA called Chargaff's experiment. And he basically figured out the rules for base pairing. A goes together with T and C goes together with G. Now, one thing you may have to do on the test is to, if you are given the amount of one nucleotide, figure out the percentage of how many other nucleotides there are. So, I'm going to give you an example here. Let's say that out of all the nitrogen bases you have, 26.5 of the nitrogen base 26.5% of the nitrogen bases are cytosine. Okay, y'all can do this on your notes or on your spare paper there. 26.5% are cytosine. I want you to figure out the percentage of guanine, adenine, and thymine. Go. Mr. Bolton came in here this morning. He said, your computer cut off? I was like, yeah. He said, yeah, mine did too. I just lost an hour's worth of work.
Nice. Was it during first period?
Yeah.
I just submitted my assignment on the computer and I pressed sign out and then it shut off. I was so happy.
It's beautiful. You got it, Walker?
We started off with,
You had 26.5% cytosine.
okay.
You people at home should be doing this too. I see you. All right, Sarah, how much guanine do we have?
26.5.
Matthew, how much adenine do we have? And thymine should be the same. So cytosine, if you've got 26.5, those base pairs, for every cytosine, you should have a guanine. So they should be the same. And then you got to add up that percent, which is 53%, subtract it from 100. And so 47% divide that by 2, and that's where you get the 23.5 for adenine and thymine. So those should be equal as well. Okay? So easy way to figure out how many of each type of nitrogen base you have as long as you have one. All right, Rosalind Franklin. the forgotten lady here. If you look at the top of 315, you can see the DNA structure where you have the sugar, phosphate, and attached to each one of those, you have a nitrogen base. You can see the adenine. Now, you'll notice that 2 of the nitrogen bases have 1 ring and 2 of them have 2 rings. We'll get into that a little bit later. One's called a purine and one's called a pyrimidine. But it's a little bit different structure, okay? Now, on 316, Miss Franklin, she performed X-ray crystallography, and she was actually the one who took the picture that helped. Who are the guys that figured out what DNA looks like? Watson and Crick. So they just figured out that DNA was the instructional unit of life. And now in 1953, they constructed the first accurate model of what DNA looked like. They wound up winning the Nobel Prize in 1962. They used the information from Chargaff's work and from Rosalind Franklin's picture that she took on 316 to determine it was this double helix structure. Now, unfortunately for Rosalind Franklin, she was using a lot of X-rays, which requires radioactive material, and in the process, she developed cancer and died. So probably she would have won the Nobel Prize along with them, but she did not because it's not awarded posthumously. And so she is sometimes forgotten and left out of the story. Now, I'm going to tell it because I think she was probably one of the most important ones in the process. Now, if you look back on 312, I think, you'll see Watson and Crick in their DNA model on that page. You can see them point to. They just look like a bunch of goofy 20, 30-year-old, late 20, 30-year-old guys playing around there. I don't know how old they were at this time. But this look like, yeah, we got that figured out. I mean, that could have been Walker and Mason right there. Who knows? Anyway, so unfortunately for Rosalind Franklin, she got cancer, died, and left out of the deal, and most people don't know who she is. So we salute you, Rosalind Franklin. Anyway, Watson and Crick figured out from the picture and Chargaff's rule that there was this double helix structure. At the bottom, hydrogen bonds hold the 2 sides together. So DNA needs to be easily unzipped so it can be what?
It can be replicated.
So it can be replicated and what else?
Synthesized.
Well, that's really the same thing. So it can be, the entire strand can be copied for DNA replication, and also it needs to easily unzip for what other process?
For coding for things. Like making proteins and stuff.
So transcription. So that a segment of that DNA can be copied in order to make proteins. What do we call those segments of DNA, Sarah? Do you know that diarrhea is a genetic disorder, Tay? Runs in your genes. All right, next section of notes, DNA structure and replication. So, we talked about the monomers of nucleic acids or nucleotides and what they're composed of. Part 2, nucleotides are linked together by covalent phosphodiester bond. I don't think you need to know phosphodiester bond. I don't know what that means, but it sounds complicated. You do need to know that these nucleotides are held together by covalent bonds. Remember, covalent bonds, what are characteristics of them?
Very strong.
Strong, they
Hold organic.
hold organic materials together. They don't dissolve in water, and they share electrons. Remember all that. The sequence of nucleotides determines what protein or enzyme is made or expressed. So whatever your DNA sequence is, that's going to determine whatever protein and or enzyme is made. Okay? Remember, most enzymes are made of protein. All right, DNA replication. When does that occur? S phase. Okay, so it's S phase of interphase. S stands for synthesis. Why is DNA copied during S phase?
So that it can just let, it has enough it has enough information that I don't know.
So it has all the stuff it needs to split apart and create 2.
Yeah, you need 2 copies of it to make more cells. So, good. That's why we're replicating DNA. Now, this process is pretty complicated and there's a lot of enzymes, so you guys need to really pay attention. Now, let's look on you see the structure on 317 of DNA there, how it's bonded together in the middle. Now, I'm going to say this. There's been some controversy recently on whether hydrogen bonds are the things that are actually holding DNA together. There's been some research that suggests otherwise. We're going to go with what's in the book, but I will try to post that article or print it off or show it to y'all tomorrow, and we could talk about it a little bit because I think that's an interesting assertion that some scientists have made that maybe that's not what is holding it together. But anyway.
Do they know what they think is holding it together?
Yeah, I can't remember what they said, what kind of forces, but I'll have to look that up. I just remember thinking, dang, that's really throwing a wrench in things. Anyway, 319, I'll have to look that one up. On 319, it talks about how is DNA replicated. So back in the day, they knew DNA had to be replicated and copied. They did not know how. And so there were basically three models that were set up. The conservative model on part A basically said that you have this double-stranded DNA structure and a copy is made from that double-stranded structure. So meaning like when DNA, like one strand went to one cell and one segment of DNA went to another cell, all of the original DNA went with one and all the new DNA went with another. Another model was semi-conservative where it said DNA split, a copy is made on each one of those sides, and then basically the new cells have half new DNA and half old DNA. And then the dispersive model said that there's some kind of mixture of the two. Some segments of DNA, each strand has a little bit of old and a little bit of new. And which one do we know to be true today? How does DNA replicate? Which one of those models? Semi-conservative. DNA unzips in the middle, spreads apart, and then you can see I've done a fancy drawing on the board over here for you three people at home. So like the black strand, that could have been the original, it unzipped, and then you have a complementary new nitrogen bases that are added in, the green strands in the middle. And so your two new strands have half old DNA and half new DNA. That is how it is copied and how it works. Okay, so let's see here. Replication fork is being done on we'll get into that in a second. In humans, it takes just a few hours to copy over 6 billion nucleotides in our cells. And if you skip down to part 3, enzymes work at a rate of about 500 nucleotides per second. So this is a very rapid process. There's actually a proofreader for this, and it's very effective and very little error takes place. So I don't know about y'all, but I can't really wrap my head around how an enzyme can add in 500 nucleotides in 1 second. Like that's some quick stuff that's going on there. And 6 billion of those nucleotides that are copied in just over a few hours. So really some mind-bending stuff taking place here. Let's go about 15 minutes. We're going to get our lab on, and we're going to make some DNA, and then we're going to look at how it actually splits. So if you flip over to 321, I'm going to pass y'all some stuff out.